Hi everyone, welcome again to the course of structural biology. We are at the end of our 11th module, protein engineering. This is the last class. In this class, because before we have discussed about what, which and how about protein engineering, the processes, rational designing, directed evolution, their details, the associated work, how the flow and all. Today I will discuss machine learning approach along with the success stories which help us understanding what is happening in this field currently and the challenges which we have to work on in future. To start, we will talk about machine learning assisted protein engineering. Any process you want to develop, if there is experimental and computational part, you always make an effort to reduce the experimental part because it is mostly time consuming, more manpower consuming, more money, a lot of setup, all are needed. So, one of the current scenario in the world is going to reduce the experimental effort using machine learning based approaches. But here we want to reduce the experimental effort associated with directed protein evolution. In addition, we want to explore the sequence space encoded by mutating multiple positions simultaneously. So, we want to make more complex designing of mutational like spectrum in each generation, and that is why we want to introduce machine learning. From our previous discussion, it is already established that screening is the bottleneck and most resource intensive step of the majority of directed evolution effort. So, if you remember, let us take one effort and we do random mutagenesis, a very common method. Random mutagenesis would include randomly the variants in the changes in the enzyme. Now, so you do not have any clue which variant content which mutation, unlike rational designing. So, how you select the better candidates or successful candidates. So, your assay is one which is most critical. So, that is why we told that our previous discussion already established that screening is the bottleneck and most resource intensive step for the majority of the directed evolution efforts. Now, devising ways to screen protein variants in silico is highly attractive. If you could correlate the design of mutants to the next step, where you could talk about the fitness, the stability and all, so that you get some selection, that would change the spectrum of how we generally approach. We could introduce much more variants, much more changes if we could have a computational screen in our hand. Molecular dynamic simulation, which we have talked about in the previous module, it is not detailed, but this will give you a basic idea that molecular dynamic simulation predict the dynamic structural changes for protein variant, and it have been used to predict changes in structure and protein properties caused by mutations. So, I have talked about, but just because we are here, suppose you see that there is a ligand and an enzyme is holding it. From the static structure, it is not possible to say how tightly the hand is holding this pen, but if I start putting something like I would hit it, so it would come in a dynamic situation, and if it is loosely bound, it will immediately open up. If it is tightly bound, it is still there. So, in that way you could understand or you could differentiate by introducing dynamics which is enabled by the process of molecular dynamic simulation. But if you want to perform a full simulation of a protein, that is also computer intensive, and you already know the huge number we have to target. So, that is definitely computer intensive. So, it requires hundreds of processor hours for each variant and a mechanistic understanding of the reaction at the hand and a reference protein structure. You need them to perform simulation. A number of other less computationally intensive physical models have also been used to identify sequences likely to retain fold and function for further experimental screening. So, we tried simulation, which is a good method, but also computer intensive, and especially considering the number of diverse mutants when you are doing directed evolution, it is uncontrollable kind of. Other less computation demanding processes are also used. An emerging alternative for screening protein function in silico is machine learning, which comprises a set of algorithms that make decisions based on data. So, I talked about few times how machine learning contributes. To introduce how it works, you could simply say they could correlate between different data set, like you have a data set A and data set B. They correlate with the values of A with the values of B. For example, if you have data here age like 10 years old, 2 years old, 25 years old, this is age data. They could correlate that with the working efficiency. Now, it apparently would be people who do not have any idea about machine learning, they would wonder how only age would correlate with working efficiency. It is not that. There are set of data like age, then some skills and all, which will be ultimately related to defining working efficiency. It is true that in many fields other than biology, machine learning is now getting tremendous success. In biology, the success is not in that measure. The reason is also understandable. If you think, I was talking about age, skill and all, they could definitely be related to the working efficiency. But so here you have different data sets and your question is working efficiency. But let us say you have skill and all those things and your question is based on honesty. The percentage of confidence you get here, the percentage would be much less, it would be a failure. So, that is a problem with the biological data. I would say there are not only one problem, but this is one of the most critical problem. Biological questions are really layered. It is very difficult to answer, that is why. One. Second, biological answers are dependent on particular protocols. For example, let us say we are talking about antimicrobial resistance. So, what we have to do? We have to take samples, do experiments, get the MICs, then get the genes involved, get their K-cats, K-ms, K-is and all. So, let us say to determine a gene beta lactamase, for predicting beta lactamase, its role along with its mutation or variability, we have MIC data, K-cat data, Km data, Ki data. You will say, what is the problem? All the data are there. Here is the problem. The problem is MIC. On which drug you are performing. Which protocol you are using. A lot of these would change the data abruptly. So, two things as I want to say, one is layered problem, layering problem, second is data inconsistency. These two have hampered a lot the efficiency of determination or getting answer in biology using artificial intelligence or machine learning. But when we come in case of protein, the structural biologists, the biochemists, biophysicists, because they work on fundamental principles, the relativeness is much less here. So, you could expect a lot data consistency here, and that is why a tremendous attention is now coming, because we already know the fundamentals of protein structure, and those information are making AI based or machine learning based prediction more efficient day by day. So, let us see the process. By building models directly from data, machine learning has proven to be a powerful, efficient and versatile tool for a variety of applications, such as extracting abstract concepts from text and images or beating humans at our most complex games. Previous applications of machine learning in protein engineering applied majorly to rational designing have identified beneficial mutations. So, we already have some success in a though little, and it was applied to rational designing because logic is there. So, we have the logic, we take those logic in the data set, we incorporate them and develop the algorithm. The practice of machine learning could possibly extend the potential of these powerful techniques, as it attractively enables rapid prediction of new variants, whereas further directed evolution campaigns may involve months of intensive experimentation. So, I have already talked about that. On the other hand, rational design often requires in-depth knowledge of the structure function relationship. So, de novo design or especially rational design need logistics. So, it needs the fundamentals. If you do not understand the fundamental, your basic is wrong, you are going to go on the direction towards failure. Whereas, directed evolution is much more a wonder, but it is simple. It is a direct relation from sequence to function, whereas for rational design you have to know the sequence, you have to know the structure, you have to know the dynamics, the energy and what not. So, at the initial level it was thought that directed evolution, there is no logic. So, how you develop an algorithm, but apparently it comes out this is a simple process. So, it could be extremely beneficial, and especially if we could correlate with the functional determination, stability determination and anything, they would be extremely helpful in reducing the number of candidates. So, reducing number of assays to actually determine the more powerful one or better ones. As the protein sequence space is astronomically large, how large it is? The number of possible protein variant for the total collection of 20 encoded amino acids of a protein having n amino acid chain length enlarges to 20 to the power n. So, you have 20 amino acids and you have n possibilities. So, 20 to the power n. Now, full sequence sampling of enzymes were regular 100 to 500, because we know 250 to 300 amino acid is average. So, you could say 100 to 500 is not achievable via any lab based or even standard computational method. So, how big is them? So, if I show you the numbers, it would be more convincing. So, for 100 amino acids, 20 to the power 100, this is the number. You could understand the enormity. 20 to the power 200 for 200 amino acids, this is the number. For 300 amino acid, this is the number. For 400 amino acid, this is the number, and for 500 amino acid, this is the number. So, I am not even saying the number, you look at the number, you look at the zeros and you will convince this is not possible for human brain, this is not possible for simple computing. So, you need prediction, you need logistics there, you need biasness, and that is why the importance of machine learning. Nonetheless, with advances in next generation sequencing and high throughput screening as well as steadily growing protein databases, the throughput in generation and the increasing amount of accessible or computable enzyme data offers the potential to exploit data driven approaches for the efficient re-engineering of enzymes. So, these I am continuously talking about in innovation or advent of NGS. What it does, I have already discussed. So, as I have given example, the price is reduced. So, you could get the genome sequence, the collection, you could get transcriptome sequence, you could get chip based sequencing, what not. You could get all possible information from sequencing, and those data along with the existing structural data around 2 lakh structure nearly is helping us to be going more towards prediction and taking help of machine learning. Based on the generation or availability of an experimental enzyme data set, statistical mathematical methods can assist in establishing a quantitative model for structure function relationship. Thereby, in the modeling one can find statistical mathematical functions that can be used to predict in silico combinatorial effects of amino acid substitutions. To perform the fitting of statistical models in an automated way, artificial intelligence and in particular machine learning are emerging as a promising solution to the challenge of efficient navigation through the vast combinatorial sequence space. So, when we are talking about data, let us think about the data driven methods. Now, machine learning as a data driven method enable exciting new breakthrough in several fields of technology and science. It became conceivable that such statistical approach could potentially come closest to an in silico approximation of the full protein sequence space, because you already have seen the enormity of the number of possibilities. So, it is thought that the only solution to that or only answer to that huge question is machine learning. This is as it allows predicting protein and enzyme characteristics and function of unknown variant of an enormous sequence space based on a comparatively small learning set of screened and sequence variant. So, as I told, a machine learning works on algorithm where data sets are there, then they understand their relation and further with the new data sets, with the set of data they could answer the question. So, they get the training. So, more and more data are coming, more and more a machine learning program would be bettering themselves. So, the betterment is going on continuously by the program. So, once you develop algorithm, you give them the initial training set, they would by their own, they would automatically understanding the correlation and developing the logic. So, more data you could make them exposed, better performer they are going to be. So, machine learning might thus dependent on the studied enzymatic property and the given input data set perform more efficient than rational protein design or unguided directed evolution experiment, because you make them, you get them, but there is no scope of improvement. Whereas, when you develop a machine learning based algorithm, you expose them to the new data set and they will do their automation of understanding. So, they would improve themselves with more exposure to the new data. Nonetheless, following an ML assisted engineering approach, an initial experimental dataset generation, thus additional sequencing of screen enzyme variant is mandatory for this case. Various data driven workflows have been developed that enabled an alternative library design that was successfully used for engineering of diverse enzyme properties, for instance to increase enzyme activity, specificity, enhance selectivity and thermostability. Typically, data driven approaches allow efficient filtering of enzyme variants by applying defined threshold of multiple filters that may, for example, distance to the active site, degree of substitution burial, or they may not, like they could not do the position specific scoring matrix or PSSM, amino acid conservation, substitution with prolines, co-evolutionary data require structural information. Data driven approaches in protein engineering commonly require data sets that map the protein sequence landscape for building predictive models. Similar to quantitative structure activity property relationship, which is QSAR and QSPR, which we use in drug development in pharmaceutical industry. So, similar to that approaches which evaluate sets of features or predictor variables to the potency of the response variable, levels for classification or regression for detecting beneficial ligand target interaction in drug designing, quantitative sequence activity modeling or QSAM for proteins were adopted. So, if you know QSAR, QSPR, they are known method where we look at the structure function of small molecule, the structure property, and then correlate those with activity. So, you could say we kind of look at features and provide a scoring or a number to them. Similar approach developed which could alternatively say as quantitative sequence activity modeling or QSAM. Consequently, QSAM was established for elucidating features that are amino acid descriptors for detecting beneficial amino acid patterns and protein variants inside a defined sequence space. So, what type of properties are there? Let us say you are looking at a protein. If you remember, the challenge here is to understand how the protein fold, and mostly the protein fold through non-covalent interactions. Very critical is hydrogen bond and salt bridge, because you could really see them. Now, if you develop a network in the protein using hydrogen bond and salt bridge, they would help you enable to make introduction of the alternative amino acids, okay, and that is what QSAM used to do. So, what you do here, you change one amino acid, like you change a serine to an arginine, and now you see the extra positive charge, extra size, how you could adopt them. This would definitely be getting help with the optimized rotamer library, which I have already talked about. For many approaches that are comparable to QSAR models, the effect of recombination is considered to be additive, and the aim of such method is to capture a general trend of combining substitutions in lieu of representing the sequence function landscape in detail. I have given the references. Similarly, the ProSAR algorithm fits a linear function based on sequence activity relationship to classify substitution, and hence identify individual substitutions that are beneficial or detrimental by deducing the independent contribution from the single residue. So, ProSAR algorithm, what it did, it fits a linear function. So, there is a linear function based on the sequence to activity, like if there is a serine, what is the activity, if it changed to threonine, what is the activity. So, this number, like if activity is 1, there should be 2, arginine it could be coming 3, in that way. So, you keep replacing and you score them, if it is better you go for them, that is the simple hypothesis. Such identified, so you get improved versions and you get the combination of the improved data set. So, those identified substitutions which are doing better can subsequently be introduced or deleted from the variant of the next round of evolution, a technique that was successfully applied for the engineering of enzymes by recombination based directed evolution. For example, improving the catalytic function by 4000 fold through the introduction of 35 substitutions to a target enzyme. However, in general, predicting the synergistic behavior of accumulating mutation is not a trivial task, especially when climbing fitness peaks with more than one amino acid at a time. So, though you are doing that, you are scoring that, but they have their culminative effect, they have their synergistic effect. When you change one, it might show one effect, but when you change two, it might totally give a different effect, and it is not a trivial task also. Also, changing amino acid, we are looking at function, but it could also introduce instability to the enzyme or protein. You have to take care of the fitness of the enzyme, and as frequently mutation can have a propensity to weak or no epistasis, as reported for the local fitness landscape of the green fluorescence protein. So, I have already talked about, so I am not introducing here because the lectures are getting long, but if you remember, I talked about the green fluorescence protein or GFP, which is a around 200 plus amino acid protein. You make small changes and it changes its fluorescence color. So, you remember, one of the major challenge here is to assay the protein, and because here your assay, if successful, gives a fluorescence color, you do not have that thought. So, a lot of work was done on GFP, and now if you see GFP was mostly popularized because of enhanced GFP. You will see YFP, you will see CFP, you will see red fluorescence protein, all type of fluorescence you will see coming from the same protein which is done by the mutations, but in some case there might be a possibility that a change of amino acid could provide a new fluorescence, but the whole enzyme become unstable, which is not normally the case for GFP protein, because if you remember, this is a beta barrel protein with extreme stability. Here, a strong negative epistasis for accumulating mutation can likely lead to each other's extinction, a behavior that could additionally be observed for a beta glucuronidase model system. Moreover, Young et al. Describe an enzyme system that revealed higher order epistatic behavior. When facing complex epistatic landscapes, machine learning is a useful method to find functions that can describe such behavior, whereas static threshold filters will likely fail to interpret. So, here I am talking about epistatic, and suddenly I remember that there would be a possibility that my young audience do not know about epistasis. So, what is epistasis? Do you know? If you do not know, please come, I will try my best to explain to you. Epistasis is a phenomenon in genetics in which the effect of a gene mutation is dependent on the presence or absence of mutations in one or more other genes, respectively termed modifier genes. So, a mutation in one gene, what that effect would be is depend on a modifier gene, that is called epistasis. I hope it is clear. In other word, the effect of the mutation of gene A is dependent on the genetic background. What that mean? That mean 10 student come to a school to learn certain things. The ability of learning would be dependent on the brain development of the earlier generation, and these are complex genetic effects. Let us say, if you see, if parents are tall, it is not guaranteed that the son would or daughter would obviously be tall. So, there are many genes which affect each other. Epistatic mutations therefore have different effects on their own than when they occur together. Originally, the term epistasis specifically meant that the effect of a gene variant is masked by a different gene. So, I am giving some example. An example of epistasis is the interaction between the hair color and baldness. Here you see two people, one with blonde hair, another with red hair. A gene for total baldness would be epistatic to one for blonde hair or red hair. The hair color genes are hypostatic to the baldness gene. The baldness phenotype supersedes genes for hair color, and so the effects are non-additive. So, if they have baldness, it would supersede the hair color gene. Another example, you will see it is a coat color genetics, mother is BCE and father is also BB. So, B dominant, B recessive, B dominant, C recessive, and then you see different combinations, B dominant, B. So, this is, when I say dominant, it means this B is for black coat, this small b is for brown coat, this big C is with pigmentation, small c is without pigmentation, which you will see here. So, if no pigments can be produced, the other coat color genes have no effect on the phenotype. So, if you have CC without pigmentation from both the side, you will not see the effect of other colors, black, brown, anything else. You will get them albino white. It is not dependent on the conditions that if they are dominant or if the individual is homozygous. Here the genotype CC for no pigmentation is epistatic over the other genes. If you see here BB, that is albino, which is for brown. When you have black and brown, they are also albino. When 2B for black coat, they are also albino and do the same. So, all the combinations, when there is no pigmentation, it dominate over and make them colorless. That is epistatic effect. Coming back, for building statistical models, an initial sequence function map can be spanned using diverse random or semi-rational protein engineering approaches. Likewise, machine learning is well suited to be applied after several rounds of established randomly or semi-rationally based protein engineering strategies, using the generated data as further input for subsequent execution of a statistically guided approach. Now, through the course of our studies, we have understood the fact that directed evolution discards information of non-beneficial variants and climbs the sequence function landscape on a single path that can be stuck in a local optimum. You understand? So, if you look at nature makes evolution, and according to Charles Darwin, it is the survival of the fittest. So, if you combine those, you will get that directed evolution goes into one track. So, there is a possibility that it could be stuck in the local optimum. ML takes advantage of all data of the sequence function landscape and employs this information to intelligently predict several recombined variants that are likely to be located close to a global fitness peak. So, the pushing from the local optima to the global optima is possible by machine learning, because it takes the advantage of all data of the sequence function landscape and then employ the information to intelligently predict several recombined variants. That push the stacking of local optimum to going close to the global optimum. Beside this, machine learning proved to successfully predict beneficial protein variants on previously identified key position, even if the prediction process was based on a relatively small input library size. Because here if you see, what you get is the training. So, that is why even from a small input library it could be enabled. A good, thus lab verified prediction can lead to an understanding of enzyme function by beneficial and detrimental variant pattern recognition, rational inspection of predicted variants, and thus the possibility of reverse engineering of enzyme variants to function. A successful ML assisted campaign for a specific enzyme might hereby enable the transfer of the developed machine learning approach to other enzymes and possibly different enzyme classes and families, allowing an innovative approach for improving and understanding enzymatic function. So, when we are trying to understand and did not know, to us it is different enzyme improving or getting evolution by nature in different way. But when we are using the fundamentals, when you are applying those fundamentals to develop a machine learning algorithm, the one successful algorithm with its individualization should work for other enzyme. That is another very critical aspect. However, such procedural workflow must not generally act beneficial for dissimilar. So, as I say the term individualization, when the enzymes are totally dissimilar, then this individualization is not easy and the procedural workflow would not work, and particularly not for distinct enzymatic properties. However, the efficiency of ML assisted protein engineering in comparison to random or semi-rational approaches were emphasized by Young, Yu and Arnold in 2019, underlining that in general the effort only increases in computational costs and additional sequencing, whereas both are techniques that highly decreased in cost during the last centuries. Nonetheless, the added cost and effort in sequencing of each single screen variant and synthesizing the predicted variant may increase the experimental load to a point where machine learning might be an insufficient approach compared to random or semi-rational approaches. In any case, it was summarized that for evolution experiments which are expensive or time consuming to screen, or when a library design needs gene synthesis, ML will likely act beneficial. Now, I will talk about some success stories in the field of protein engineering, how it worked, what it happened. I would also talk about some labs, some initiatives which are instrumental in pushing the journey of protein engineering. I have already talked, but computing methods have been used to de novo design of a protein with a novel fold named top7. You all have remembered in our previous classes we talked about that. Top7 is a protein designed in David Baker lab with the help of the Rosetta software. This is the first protein which was expressed, and the determined structure clearly established the fact that in nature we do not have such fold. This is a landmark. There is also example of development of novel sensors for unnatural molecules. I have already shown that how alpha helices are taken and then dimer, trimer, tetramers are made, and thus again the structure solution clearly shows that they have adopted very different conformations. The engineering of fusion proteins has yielded rilonacept, which is a pharmaceutical that has secured FDA approval for treating cryopyrin associated periodic syndrome. So, this is one of the success in medical science. Another computing method, IPRO, successfully engineered the switching of cofactor specificity of Candida boydini xylose reductase. So, little bit more on IPRO. Iterative protein redesign and optimization redesigns protein to increase or give specificity to native or novel substrates and cofactors. This is done by repeatedly randomly perturbing the structure of the protein around specified design position, identifying the lowest energy combination of rotamers and determining whether the new design has a lower binding energy than the prior one. So, what it do? It is iterative protein redesign and optimization. So, there is a iterative method, that is why the computer algorithm introduces it. Initially does the change. So, design, then redesign and then optimization. So, how it is performed? So, firstly it redesign protein to increase or give specificity to native or novel substrates and cofactors. This is done by repeatedly randomly perturbing the structure of the protein around specified design position. Suppose you have this protein and this is where you think the residues which are used for. So, you call it active site, let us say. Then you make changes and then, so you make variants in those positions, identify the lowest energy combination of rotamers of those amino acids and then determine whether the new design has a lower binding affinity or binding energy than the prior ones. Some more about IPRO. IPRO offers an integrated environment for altering protein binding affinity and specificity, which we talked about, grafting a binding pocket into an existing protein scaffold. So, you have one protein with its binding pocket, you have other protein, you graft this from these two here. Predicting an antibody's tertiary structure based on its sequence. So, sequence to structure prediction, enhancing enzymatic activity the same way, assessing the structure and binding energetics for a specific mutants. So, what are the applications of IPRO? Structure based computational enzyme design, which we talked throughout, designing novel antibody binding pockets, altering enzyme cofactor specificity. So, all of these are done by identifying one part, then keep mutating, optimizing rotamer and look at the entire set of combination is providing a betterment or not. A computational procedure for transferring a binding site onto an existing protein scaffold, an iterative computational protein library redesign and optimization procedure, protein library design using scoring function or class maps. When you are developing variants, let us say you have a protein, here is the active site, here is a serine molecule, this is a serine, this is a let us say histidine and this is a aspartate. So, now you keep changing them. So, change 1, change 2, change 3 and all, similarly here, then make those combinations and see that when you make them, are they make any steric class or not. If they are making steric class, then take them out. Also, you develop a score so that your changes would better or not. Modeling and optimization of directed evolution protocols and pore designing, because all of these as a sentence, as a word makes sense to what we are talking about, but pore designing is something different. I would talk about that. So, pore designer is a IPRO suite program, it is a subset of the IPRO program. What happened? Pore designer was successfully developed to redesign bacterial channel protein OMP-F. You all know about OMP-F. If you do not know, this is a channel protein, but here the pore designer program trying to reduce the 1 nanometer pore to sub nanometer dimension. So, this is a 1 nanometer pore, 1 nanometer 10 angstrom and you want to go narrow. The strategy is to change with many amino acids and it was found that tryptophan in both the places will be stable there and make the pore narrower. So, 2 mutations from pore designer predicted in silico redesign of OMP-F protein, the aspartate 113 tryptophan and serine 125 tryptophan mutations resulted in a smaller pore lumen and more hydrophobic inner pore wall. So, what they did, they have identified residues around, they keep them mutating, they optimize the rotamers and then calculate the energy so that the mutations are stable or not. If stable, then what are the size of the pores. The wild type residues have been shown in green and the altered residues have been shown in pink sticks, as you could see here. Transport experiments designed pores reveal complete salt rejection when assembled in biomimetic block polymer matrices. So, their goal was to make differentiation. So, what they did here computationally, now they come to experiment, they perform transport experiment and the mutated or altered OMP reject the salt, which makes the process, which clearly shows the design is successful. Computer aided design has also been used to engineer complex properties of highly ordered nanoprotein assemblies. If you do not have idea, I have talked about this. So, nanoprotein assembly, it comes from the inspiration of DNA origami. DNA origami is a field where a lot of DNAs fold together to develop a kind of 2D, 3D structure, and that is used here. I talked about the story of using the principle of DNA origami in Baker's lab. Similarly, a protein cage, which is E. coli bacterioferritin ECBFR, which naturally shows structural instability and an incomplete self assembly behavior by populating two oligomerization states, is a model protein for this study. Through computational analysis and comparison to these homologs, it has been found that this protein has a smaller than average dimeric interface on its two-fold symmetry axis, due mainly to the existence of an interfacial water pocket centered on the two water bridged asparagine residues. So, they were instable. So, when you did, as I told, in designing, you have to understand the fundamental principle. So, when they have done the analysis, they found that the protein has smaller than average dimeric interface. To investigate the possibility of engineering ECBFR for modified structural stability, a semi-empirical computational method is used. If you see, this is the typical structure of octahedral maxiferritin and miniferritin. Maxiferritin is 1 BFR, 1 BFR is the PDB ID and 1 DPS is the PDB ID for miniferritin. They are structured with 4 helix bundle monomer. If you see here, the A helix is orange, the B helix is yellow, the BC loop in maxiferritin, this BC loop is cyan. Here you see the BC is a kind of 310 helix, the C helix is in blue, the D helix is green and E helix is red, which is present in maxiferritin. Attempt was made to virtually explore the energy differences of the 480 possible mutants at the dimeric interface relative to the wild type ECBFR. This computational study also converts on the water bridged asparagines. Replacing this to asparagine with hydrophobic amino acid results in protein that fold into alpha helical monomers and assemble into cage, as evidenced by circular dichroism and transmission electron microscopy TEM. So, they performed CD and TEM experiment. Both thermal and chemical denaturation confirm that all redesigned protein in agreement with the calculation possess increased stability. So, they were able to enhance the stability of the natural protein. One of the 3 mutations shifts the population in favor of the higher order oligomerization state. If you remember, they have a dimer state. So, it pushed to the higher order oligomerization state in solution, as shown by size exclusion chromatography ACC and native gel or Tris gel electrophoresis, because when you run normal gel electrophoresis, SDS electrophoresis, because of the presence of SDS, because of boiling the condition, because of adding of the dye and all, the protein goes to the original monomer form. every protein exists in their monomer form. If they are oligomer, it breaks because it is through non-covalent interaction. That is why they have run the native gel electrophoresis. I will talk about Institute for Protein Designing in University of Washington. This is the Institute for Protein Designing. This guy is David Baker. I have constantly talk about. He have taken a instrumental step in developing protein design principles. So, as I was talking about, not a lot of groups around the world have taken the initiative, because if you see, it is not about one scientist, it is about a group. You see, the one building is dedicated, and you see the organization structure: Trisha Davis, PhD Chair, Department of Biochemistry; David Baker, PhD Director; Lance Stewart, PhD MBA Senior Director of Strategy, are the part of the organizational structure, but then there are many scientists, they are working together. And how big the initiative, you will get an idea looking at the funding organizations: Bill and Melinda Gates Foundation, Defense Threat Reduction Agency DTRA, Defense Advanced Research Project Agency DARPA, Department of Energy DOE, Life Science Discovery Fund LSDF, National Institute of Health NIH, National Science Foundation NSF, Takeda Pharmaceuticals, Washington Research Foundation, and beside them a lot of individual individuals are contributing. So, yes, protein engineering is our dream. There are a lot of people who are capable of, but it needs an initiative where all the experts would come into one platform, work together, facilities would be in-housed, and all these are challenges to many developing countries like ours. So, the executive function of naturally occurring protein is to solve the challenges faced during evolution. However, Institute of Protein Design, or IPD, they face challenges that are not faced during natural evolution because of the current situation. The goal of the Institute of Protein Design IPD is to develop and apply method for designing a whole new world of synthetic proteins to address these challenges. So, this development by itself is a great achievement. To achieve the previously mentioned goals, what they did, the Institute for Protein Design was established in 2012. As I told, this is a very nascent field, this is a very young field. So, the first kind of initiative was developed on 2012. Protein design require high level expertise and talent in computing and software, biochemistry, genome science, biological structure, pharmacology, immunology, and other basic science disciplines, as well as clinical medicine. In one word, a completely extremely interdisciplinary setup which they have got there. It is an initiative to build up a wonderful team using faculty, scientific staff, postdoctoral fellows, and graduate student in University of Washington, as well as developing partners from collaborating institution, innovator networks, and from the computer and biotechnology industries, bringing extraordinary expertise to bear on a singular focus to advance the potential of protein design. Over the past 16 years, University of Washington researchers have made significant progress in protein design and protein structure prediction, developing the world leading Rosetta software, which I talked earlier. Over this period, those scientists have developed methods for designing protein with a wide range of new functions, including catalysts for chemical reactions, HIV and RSV vaccine candidates, and flu virus inhibitors. The IPD integrates the strengths in protein design with Seattle area expertise in biochemistry, engineering, computer science, and medicine, and leverages the exceptional Seattle strength in the software industry. So, IPD looks for 5 grand challenges. They are working on building better vaccines for pathogens such as the flu. They combine the protein with nanoparticle to and make many designing so that they could develop better vaccines. This is very interesting. They are going to expand beyond nature's alphabet of 20 amino acids. So, if you remember, you just do the mathematics, 20 to the power 100, you see the number, and then do 21, 22, 23, and you see the increase with increase of even one amino acid. That is what they are doing. They are taking unnatural or non-natural amino acids. They are creating advanced protein containers for targeted drug delivery. So, you have target domains, they take them so that they could target a particular region and then fill the inner with drugs which they want to be delivered there. Designing smart protein system that can identify cancerous or otherwise unhealthy cells. So, you have to identify. So, they have switches where you keep the interactions. So, you have the target which is inactive, then you have the switch with induced key, the switch would be activated. For example, let us say you have a interaction and that interaction like a phosphorylation with a kinase activity, this will interact with and then you get a fret signal in that way. Then another very interesting aspect is making next generation nanomaterial for use in solar energy capture, nano fabrication and many things else. So, you could see that they have already developed, like if you see dimer, trimers of proteins are there and they are used for development of those next generation nanomaterial. Few success stories from David Baker lab and this protein design initiative. Ultra potent vaccines. In 2010, researchers from IPD demonstrated that Rosetta designed protein. So, when I say IPD, you might wonder that IPD was in 2012. So, why I say 2010? It is the continuation, the people who have now assembled to IPD, I am talking about their success. In 2010, researchers demonstrated that Rosetta designed protein can take on shapes that elicit neutralizing antibodies against HIV when injected into animals as vaccine formulation. 4 years later, 2014, they have applied the same method to create novel immunogens against the respiratory virus RSV, a particularly lethal virus for infants and the elderly. In 2019, they reported their first fully synthetic nanoparticle vaccine targeting respiratory virus RSV. These and other vaccine candidates are now being developed by Ecosavax, a IPD spin-out company. So, they are not only doing research, they are converting those research to translation, they are developing a lot of spin-outs. In 2020, IPD used this same platform technology to create ultra potent COVID-19 vaccine, that is animal testing elicit neutralizing antibodies at levels more than 10 times greater than the antigen from authorized mRNA vaccines. Human clinical trials are now underway. Cancer immunotherapy. In 2019, IPD created compact proteins that stimulate the same receptor as IL-2, a powerful immunotherapeutic drug, while avoiding unwanted off-target receptor interaction. So, they make the natural attack, the natural immune more specific and get rid of the side reactions. These synthetic proteins shrink tumor in mice already. This technology is now being developed by IPD's spin-out company Neoleukin Therapeutics as a safer platform for cancer immunotherapy. Rapid antivirals. In 2011, IPD designed synthetic mini proteins that target conserved surfaces on influenza hemagglutinin from the 1918H19N1 pandemic flu virus. These durable antiviral proteins protect rodents from exposure to lethal amounts of the virus. Researchers from IPD have also shown that the same flu binders can be used as diagnostic reagent in low cost diagnostic test strips, providing improved performance as an influenza assay compared to a traditional antibody based capture system. This sensitive reagent can reliably detect less than 100 influenza virus particles on a single nasal swab. In 2020, they used this same platform technology to create potent antivirals for the pandemic coronavirus and adapted them into modular and sensitive biosensors. Celiac disease. In 2011, a team of University of Washington undergraduates working out of the IPD won the grand prize at the annual iGEM competition. If you guys do not know about iGEM competition, it is a competition of mostly undergrad and master students. In each institute, the student of especially from life science background, biotechnology background, they assemble, they get a plan, they develop a model through computation, and if possible, they by taking help of the research groups in their institute, they make some experiments. This is something which have make a lot of young generation interested towards genetic engineering related research. So, coming to the story, the team won in 2011 the iGEM competition. They sought to develop a cure for celiac disease and used computational design to re-engineer a natural enzyme to break down gluten in the harsh acidic condition of the stomach. In 2015, IPD team published the most advanced version of the enzyme dubbed Kuma O030, which clips gluten molecule at sites that are known to cause an immune reaction in those with celiac disease. The enzyme was further developed by IPD's spin-out company PPP Biologics, which has been acquired by Takeda. Immune silencing. In 2014, IPD scientists were the first to demonstrate that computational design can be used to eliminate T cell epitopes from proteins by altering the amino acid sequence without affecting structure or function. The immune silencing approach enabled collaborators at the National Cancer Institute to improve an immunotoxin used to treat cancer. The new designed immunotoxin maintained good activity, stability and anti-tumor activity. Their reduced immunogenicity will enable more effective cancer therapy as more treatment cycles can be given before an immune response is mounted to neutralize the therapy. So, these are few success. Tale of heme enzymes. If you remember, while going through directed evolution, I was talking about things when you have the enzyme, you do engineering towards what? You have the enzymes reactivity A to B, you want like it would react to A1, A2, A3, A4, A5 to produce B1, B2, B3, B4, B5. So, the related substrates. So, the enzyme is working on A, you want it to work on many more, so that you could expand the chemical repertoire of the enzyme's transformation. Once it was successful, then people were even more imaginative. They want to bring a totally different substrate and want to make the transformation. Taking heme enzymes, especially which are monooxygenase, monooxygenase work on CH bond, one of the most stable bond and one of synthetic chemists life. So, they all the synthetic chemists, a lot of them are invested their labs energy on this. But if you have these heme enzymes, they are all actually working on them. When they are working, they are very specific. So, the departure, as I told, CH to COH, C double bond C to epoxide, R2S to R2SO, R2SO2, RCOH to RH plus CO2, RCH to CH2, R prime to RHC double bond RCH prime. So, many reaction, but then now we are going from there to abiotic reactions. Abiotic. What is abiotic? Think about, it is doing CH conversion, but mostly biologically relevant reactions. If we could make biologically irrelevant, but which are synthetically required, then this could open up a new era. So, exploring the engineered enzyme catalyst towards transforming new substrates or newer set of transformation, especially the abiotic reaction, opening the new world of possibility. A heme protein was viewed as a self-assembling, fully DNA encoded catalyst whose electronic and static properties can be tuned by the help of evolution. It is an iron complex that can be modified simply by mutation of the DNA to make it capable of all sorts of very interesting chemistry. Enzyme P450 variants are used to make in a single step the development of cis-cyclopropane, which is the core of the FDA approved drug Fedzima. Wonderful selectivity was achieved for a single stereoisomer, making the conversion process easier and, more importantly, cheaper. Another example of great selectivity is the ability to make the key trans-cyclopropane intermediate to another drug tisagrelor. A single biocatalytic step at room temperature in aqueous solution replaces 5 or 6 chemical steps, often involving a precious metal, and provides an environmentally friendly way to make this design. So, I discussed this about, but just to end up with, when enzyme do it, it would do physiological condition 37 degree centigrade, normal pressure, no metal catalyst, and mostly one step. So, these 4 are good enough: one step, normal temperature, normal pressure, and no metal catalyst is making your life much easier. Along with that, the specificity. So, this is really good. Coming to new approaches and future directions. Even though designing a protein remains a challenging task due to the large sequence space that requires sampling, few landmark development have changes the way how we looked at it. The number of gene sequence enhanced astronomically following up the success of Human Genome Project in 2003. Next generation sequencing techniques are not only providing us the sequence of a gene or protein, it also providing the genetic regulation, expression patterns, effect of factor influencing gene, protein expression. All of these are invaluable in correlating structure to function, further contributing to protein design. Though much smaller in comparison to sequence, the number of resolved 3D structure also are increasing day by day, thanks partly to Structural Genomics Consortium. Apparently, looking at the available data, it is not uncommon to consider that I already have talked about structural genomics. So, it is, we all, even as a structural biologist, we realize that, or we feel that structural genomics project did not go and achieve the success the human genome project have achieved, but in reality this initiative have provided us with many things which helps new generation protein crystallographers to work with much larger scale. To name few: new crystal screens, automated solution makers, crystallization robots. You see, these are the crystallization robots. There are two type of, one is for, if you look at hanging drop and sitting drop, both automation are possible. Automated crystal checker. So, if you see like this, you store your plates, and the before you store you have to record them. There is a microscope, there is a robot which will take the plates, check and take its photo and organize them properly. You come after 7 days and keep checking all the conditions. So, that is really good. Microfluidics and nanofluidics based crystallization setups, they have also contributed hugely. I also have talked about them. This is further added with exciting development in the field of cryo-electron microscopy, helping the community to be awarded the Nobel Prize in 2017 and to be nominated by Nature as Method of the Year in 2016. Cryo-electron microscopy has traveled a lot, which I have told in the module of cryo-electron microscopy. If you see among the structure solution techniques, this is the most upcoming one, because it have ability to get or solve the structure in more native state. It could work with bigger size complexes, which is what we are looking at. So, definitely the advancement in cryo-electron microscopy is a great thing towards understanding biology, especially the complex interactions and further protein designing. Increase in efficiency and automation enhances the number of solved structure entry in the PDB database. Those increment enhances the efficiency of a number of algorithms that use sequence and structure database to learn various sequence to structure features. Needless to say, machine learning and deep learning neural networks are emerging as key players in this domain. We have already discussed about them. Cadet et al. Came up with a supervised learning of enantioselective selective enzyme sequences and activity of n individual point mutation to predict the activity of all combination the twin of this point mutation. The method involves numerically encoding the sequences, wild type and single mutants and experimental activities, converting them to a signal using Fourier transform and using a partial least square step to predict the activity of a mutant, which is a combination of multiple point mutation fed in the learning step. The correlation coefficient between 28 mutants validated experimentally revealed a good agreement. The non-conventional crowd-sourced outline competitive gaming protocol Foldit 180 to use human intuitions as a lever for accessing novel catalytic folds or predicting folded polypeptide geometries. So, I talked about this Foldit, which is a part now of Rosetta Common, basically an initiative initially taken by Bijay Pandey, but now they have joined hand with David Baker. What happened? They ask people to form games, like form teams and play games where they are making permutation and combination of different non-covalent interactions to optimize the fold of a protein. Factoring in contribution from binding pocket geometries, alternate catalytic motifs and hydrophobicity of the pocket would be a step forward in using these algorithms more reliably. Popova et al. Have developed a deep reinforcement learning tool for drug discovery to identify molecules with desired properties such as hydrophobicity, melting point and inhibitory activity against specific enzymes. Instead of constructing novel small molecule libraries, if this workflow can be used for screening whether a ligand will show activity against a library of an enzyme and its mutants, this could emerge as a useful enzyme engineering tool, which is yet to be established. Protein design thus remains an active field of research for the search of a unified set of rules that can be used for tuning substrate and cofactor specificity and tailoring novel functionalities or redesigning them anew. It could also be worth mentioning that directed evolution and computational design have also been aimed at creating synthetic pathways that take advantage of the new enzymes along with several updated genome scale networks of eukaryotes and pathway redesign tools. So, initially where we are working on enzyme or protein, now we are working on enzyme pathway, what is metabolic pathway engineering. So, we are going into the hierarchical state. The marriage of new algorithms and directed evolution approaches bears promise of generating efficient catalysts needed by the food, pharmaceutical and renewable energy industries. As I told, this is definitely the future. Last but not the least, predicting protein structure with accuracy comparable to experimental techniques. The main metric used by CASP, you already know what is CASP, to measure the accuracy of prediction is global distance test or GDT, which ranges from 0 to 100. In simple terms, GDT can be approximately thought of as the percentage of amino acid residues within a threshold distance from the correct position. So, remember, when we compare between two proteins, we use RMSD. The GDT could be correlated or comparable to RMSD. According to Professor Moult, a score of around 90 GDT is informally considered to be competitive with result obtained from experimental methods. So, GDT is measured in from 0 to 100 if a prediction program could score more than 90, the prediction would be considered as per experiment. In the result from the 14th CASP assessment released last year in 2020, latest AlphaFold system achieves a median score of 92.4 GDT overall across all target. This is amazing because first time a program showed that their continuous prediction is comparable to experiment, and it is not for one structure, it is the continuous performance. That is why it is very important. This means that the prediction of an average error of approximately 1.6 angstrom, which is comparable to width of an atom. You understand? So, 1.6 angstrom could be a RMSD for a very similar protein. Even for the very hardest protein targets, those in the most challenging free modeling category, AlphaFold achieves a median score of 87 with a very difficult targets. So, this is the CASP and the performance of AlphaFold. You see 2018, because it comes in every 2 years. So, this graph shows 6, 8, 10, 12, 14, 16, 18, 20. We will get the next one in 2022. If you see, this is around 92.4% or 92.4 GDT. And when you see this, this will also be interesting. Here you see that the green part, the green structures are experimental and the blue is computational, and you see how beautifully they agree to each other. A lot of people, especially my students whom I used to teach about the protein folding problem, I talked about the Anfission's experiment, Leventhal paradox, and how we are investing a lot of research group, their continuous research to understand the fold, especially the undergrad students. They all mail me, especially my ex students, a lot of them mail me, sir, so the problem is solved now. So, is it the end of experimental process? So, what I did, I laughed, because yes, this is a very exciting thing, but It is just opening up the potential for biologists to use computational structure prediction as a core tool for protein or enzyme designing. Further improvement of machine learning methods may prove especially helpful for important classes of proteins such as membrane proteins that are very difficult to crystallize and therefore challenging to design and modify. So, this will bring, if you remember, there is a huge gap now between the sequence and structure. These will be very helpful in making this gap narrower. Now, a lot of people ask me what is my dream as a researcher, as a scientist. So, I am taking the help of Nobel laureate Francis Arnold's ideas to answer this. If we could rewrite the code of life or even just the code of enzymes, then we could look at an organism, say a yeast or a bacterium, as a chemical factory that uses renewable resources and convert those valuable products all programmed in the DNA. That is the dream. There is the ultimate programmable green chemical synthesis machine. There is no waste, especially no harmful waste. Every waste will be converted to valuable product. It has been hard to implement though, because it is very critical to make the desired modified microbes if you want to change an entire microbe, because nature have developed a lot of restriction, so that it is difficult to modify the entire them. And instead of making an altered microbe, easier initial step that we want to make the chemicals that we want using protein or enzyme. So, instead of making a modified microbe, let us dream about making the enzyme, modified enzyme or modified protein. That is a very difficult DNA design problem for sure. Progress in creating new enzyme has been stymied by two major hurdles on the path between a DNA sequence and what the resulting protein does. The first one, how does the sequence encode a folded protein? Fortunately, standing there in 2021, AlphaFold have shown a light on that front, and that brings us to the more important second question. The more important issue is how we could convert those new designing to desired function. The fate of human civilization would be heavily dependent on our success in getting answer to that question. So, with that, I hope you understand the importance of protein designing, how the entire learning of this course is coming and applied to this, and that is the beauty of basic science to application. Protein engineering, enzyme engineering have given us a platform to utilize the knowledge we gather through our PhD postdoc training, learning molecular biology, learning how to clone, how to synthesize, how to understand gene, how to work with the protein, how to characterize the protein, how to make mutants and all these things, and then how to solve a high resolution structure, how to use a lot of techniques, spectroscopy techniques, microscopy techniques, chromatography techniques and all to get a pure protein and make their characterization, and the training of functional assays. All of them come together to develop or to help us working on a applicative area like protein engineering or something which is structure based drug designing, which I will talk about in the next module. Thank you very much for listening. Please keep listening and keep asking questions if you have any doubts. Thank you very much.